we're working on a worksheet, and what I want to look at with that worksheet today is the bottom questions on endo and exocytosis. So we talked about the passive transport. Now we want to talk about active transport. So have that worksheet handy. We are going to look at, so I'm going to show a video first and again, have the worksheet handy so that you can look at those pictures at the bottom of the worksheet so that you get an understanding of what that means. All right, let's see. And I got 2 people who have turned it in already, but you still need to pay attention because just in case you didn't do it right, you need to make sure that, oh, let me try something else. Hmm.
This video is on endocytosis and exocytosis.
Okay. Oh. Okie dokie. All right. Found a different way to pull up the video. All right, because I had already included this on your playlist. So that I decided that's a better way to look for it. Okay. All right, so again, we're going to look at active transport today. We did everything, then we're going to review it all. Just keep in mind that your exam, I think 4th period is next Friday, so you have the last exam. So we have a extra minute to review. So so those of you, like I said, there's 2 of you who've already turned in your worksheet. Please don't tune out thinking I'm done, because you might not have answered the questions correctly. So please make sure that you pay attention. Oh my goodness.
Endocytosis and exocytosis. The cell membrane is a phospholipid bilayer. Stuff moves across the membrane to either get into the cell or to get out of it.
I paused it there because this section is on the test, so let's consider this the review for that. So you need to know the cell membrane is a phospholipid bilayer. What does that mean? It means that there's 2 layers. Bilayer means there's 2 layers of those fatty acid molecules, those phospholipids. We learned that on our macromolecule chart, okay? So we're tying things together.
Small things like ions, they can use protein channels and they can diffuse. But what about macromolecules? They need another way, and that's by using vesicles. These vesicles pinch off the membrane. If they're going to bring things into the cell, the process is endocytosis, because endo is internal. And if instead the cell is throwing something out, that's by exocytosis. Exo is external. And these are the methods of bulk transport. There are different kinds of endocytosis. It could be phagocytosis, pinocytosis, or receptor-mediated endocytosis. Phagocytosis means cell eating. This happens only in a few kinds of cells, like in the immune system, neutrophils and macrophages. What do they eat? Pathogens like bacteria, tissue debris, etc. Whatever the substance may be, when the cell comes in contact with it and it wants to engulf it, it will form these extensions called pseudopods. They go around the substance, meet, and fuse. Now it gets pulled into the cell. and separates from the membrane.
All right, I'm gonna pause that right there. All right, so this is a vocabulary word, pseudopods. Pseudo means false feet. Okay, so pseudo anything is false, and pods are always going to be feet. In this case, it's going to be like more like fingers and not feet. But what it's saying is the cell membrane forms a pocket, and the pocket kind of has it's going to grow towards each other. So eventually, see the arrows, those two little extensions of the cell membrane are going to join, and it'll form a vacuole.
Now this cavity, lined by that phospholipid bilayer, is called a phagosome, and it's got the engulfed material inside. That needs to be broken down. Among the organelles, which one does that? The lysosome. The phagosome fuses with the lysosome to form a phagolysosome. These lysosomes have hydrolases. They will digest the material, and what remains that's undigested, that cannot be broken down, that forms the residual body, which will then get extruded from the cell. This extrusion is by exocytosis, because the cell is throwing its contents out. So that is phagocytosis. Pinocytosis is similar, but it's different. Instead of eating, the cell is going to drink now. This is fluid phase endocytosis. It gulps in the extracellular fluid with its dissolved solutes. So the vesicles here are smaller. These are called pinocytic vesicles, or pinosomes. They will then fuse with an endosome for further metabolism. Unlike phagocytosis, pinocytosis is seen in most cells, for example in the small intestine. But it's very non-specific. The cell is just gulping down the ECF with whatever is in it. This process can be more refined with receptors, and that's what happens in receptor-mediated endocytosis. Now, most forms of endocytosis use something called clathrin to form the vesicles. This clathrin has a triskelion structure, so it has 3 legs. It's a 3-dimensional structure, so it forms a cage around the vesicle. Receptor-mediated endocytosis usually involves clathrin, and if it does, it's called clathrin-dependent endocytosis. The cell membrane has depressions. Clathrin is located beneath these pits. These are coated pits. They are coated with specific receptors. The two are attached by things called adaptins. So when the ligands bind to the receptors, the vesicle is going to form. It's more specific than fluid phase endocytosis because a specific receptor is involved. Now, as the vesicle forms, there's another molecule called dynamin. That's located at the neck of this vesicle. It's a GTPase, so it uses GTP and helps pinch the vesicle off the membrane. This vesicle then loses its clathrin coat. So that's uncoating, which is done by an uncoating ATPase. So it uses ATP to remove the coat, and the clathrin can be used to form another vesicle. After uncoating, it fuses with an endosome to form an early endosome. The endosome has proton pumps on its membrane. They send hydrogen ions into its lumen, keeping the environment acidic. That acidity helps the ligand detach from its receptor. So it's like a sorting center. The receptors are going to get recycled back to the plasma membrane. What's left behind are the ligands in the endosome, which is now going to form the late endosome. That late endosome will fuse with the lysosome. The lysosomal hydrolases break down and digest the content. A classic example used for receptor-mediated endocytosis is with low-density lipoprotein receptors, which take up LDL cholesterol from blood. It goes through this process in the cell and gets broken down to cholesterol. So that's just an example of receptor-mediated endocytosis. Clathrin is the most studied in endocytosis, but there are clathrin-independent methods as well. For example, caveolae in endothelial cells. So there's lots of different kinds of endocytosis, but all of it would involve things entering into the cell. What about leaving the cell? What would the cell want to throw out other than waste material? Proteins, hormones, neurotransmitters, things like that. Stuff that it synthesizes and has to leave the cell and go somewhere else and perform its function. Proteins get synthesized in the cell and then they go through all modifications through the endoplasmic reticulum and then through the Golgi apparatus to get packaged into vesicles. If the content of these vesicles has to leave the cell, it will do so by exocytosis. There are different pathways for that, like there's the constitutive pathway, where when it's synthesized, the content gets released. There's no storage. These are called secretory vesicles. The second type is regulated, where the content can be stored, such that when there is a signal, it can get released into the extracellular space. An example of this is at the synapse, like the neuromuscular junction. The neurotransmitter is stored in vesicles, and when the action potential arrives, calcium enters the cell and makes the vesicles fuse with the membrane. The fusion of the vesicle to the membrane involves different proteins. One complex is the V-snare T-snare complex. V is for vesicular and T is for the target organelle. Along with this, there are other proteins and GTPases. Now this complex, it helps with docking of the vesicle onto the membrane so that the two can fuse and the contents of the vesicle can be released. There are actually different ways that this could happen. One would be where the vesicle briefly opens, releases what's inside, and then closes, so it can be recycled and used again, which is one of the ways that exocytosis happens at a synapse. Another way would be complete fusion with the target membrane, and then releasing the contents of the vesicle. If it does fully fuse, in exocytosis the cell gains membrane, and in endocytosis a bit of the membrane gets back into the cell. The two thus couple, so the cell size stays the same. And that is endocytosis and exocytosis. If this video helps you, give it a thumbs up and subscribe to my channel.
So
Thanks.
that is on your playlist. So if you need to look at that again, you can do that. All right, so what we are looking at completing today. So we started yesterday. We made sure that we answered the top question. So each of these columns should have 2 check marks, and you somewhere you should have drawn a picture. Malaysia, you were absent yesterday. So this worksheet is, let me come out of this for a second. All right, you should have Actually, this is in the module. This one is in the module. All right, so we are in the module for this one. And this is cell transport. All right, so that's where you find that. Yesterday, we drew some lovely pictures so that we know what we mean by, we use the red blood cells as our examples so that the pictures are like, I thought they were on here. Okay, I know where they are. They are in your module. All right, so the pictures that we drew yesterday, so that we can properly, so this is what we're looking at. We did passive transport yesterday. Today's discussion was about active transport, where we actually had to use some energy to get molecules to go in past the membrane. So the video talked about pumping things in, exo, and again, exo, think exit. All right, things have to leave the cell. Endo, think into. Things are going into the cell. All right, look at these vocabulary, active transport. The molecules are going from low to high. Remember, the concentration gradient when we're talking about passive transport is going from high to low, where there's a lot of molecules and they're trying to spread out, so they're going to an area where there's low. This time with active transport, we need some things that are outside the cell and they might be in low concentration out there. So we have to use energy, and ATP is your energy molecule, to bring that molecule inside the cell. Okay, so it's going to go the reverse of the concentration gradient. It's going to go from low to high. All right. That picture is on here. I think it's on here, but you got to go through all the stages.
Observing diffusion.
All right, so Malaysia, we went through all of this yesterday.
Tonicity is the measure of how much water versus solute there is in a solution.
All right, that's the picture I'm looking for.
When animal cells, such as red blood cells pictured
All
here,
right, so I made sure everybody drew these cells. If you didn't draw it yesterday, you should have. And this picture is what the worksheet is referring to, is what are the cells doing? All right, so we're in a hypertonic, and again, folks, if y'all were here yesterday, this is a review. Hypertonic, the cell is going to shrink because all the water is coming out. In an isotonic, the cell is going to remain normal because you have an equal amount of water going in as coming out, so the cell stays nice and round. Hypotonic is going to burst or swell. Swell is the word that's on your worksheet because everything is going in, nothing's coming out. So it's going to swell. If it can't regulate it, it will burst. Okay, so all of that was with our passive transport. So today we were looking at defining active transport. We looked at exocytosis, where the process of releasing molecules, so things are exiting the cell. Endocytosis, it's the cell is taking things in, and sometimes it actually has a pump where there are transport areas on the membrane that are there in order to actually pump things in and out of the cell. All right, so we're going to go back to the worksheet. All right, so that's, again, each of these up here at the top should have two check marks. Then you're going to take these letters based on the vocabulary and the discussion from the videos and match these up. All right, for this part right here, and I think those of you, the two of you who submitted, I commented on, and both of you need to look at those again because not everything is correct. You've got 4 words here that need to be placed next to each of these pictures. You can repeat the word more than once. So you're looking at the law of diffusion, things going from high to low. Gases can diffuse. Water diffuses. Big molecules, proteins, fats, lipids, those types of things, those need to be actively transported across the membrane. So then you're looking at that in terms of using, is it diffusion, active transport, or osmosis, or equilibrium, meaning nothing is, the cell is not bursting, it's not shrinking. What's going on? So you want to put one of those 4 words next to each of those pictures. Then you want to, here again, you're looking at the concentration inside the cell and outside the cell and trying to decide which way the water is going. And is the cell, so you're circling or highlighting one of these words. These are fill in the blanks, but there's choices. So you're choosing a choice. On the first one, will the water flow into the cell, out of the cell, or in both directions? So will the cell burst or stay the same or shrink? Next one, the same thing. You have choices. So you want to circle or highlight one of those choices. All right. And this goes with the so if you have trouble with question number 4, go back to that same little section that we were looking on in Canvas, or go back and look at the video that we looked at yesterday. Again, that little red line is called the concentration gradient, but it's supposed to represent the cell membrane. So you're looking at which molecule, how many molecules, how many red dots are on one side and how many are on the other side, and look at the arrow. So the arrow is worried about the blue dots, not the red ones, about what the blue dots are doing. And the blue dots are water. So what is going on here? All right, using the transport protein to move things across. So what is that? What is it called? If you're going to use a transport protein to move things across the membrane, what is that called? All right, and down here, this is describing what we were just watching in the video. This one, it's eating something. This one, they're showing you follow the numbers. You have the thing inside. Next, it goes to the side and it spits it out, and then the membrane goes back to normal. What is that called? All right, the last one. So all three of these have to do with active transport. Then you had 3 words. So put those 3 words in the correct place down here. Okay. All right, I'm going to stop talking and let you finish working on that. That's due today. That's due today. So when I see you again on Friday, we're going to look at reviewing. Okay, we will close this, go back, go back. All right, so if you finish that, you want to come back and look at before you go and be able to, this is a you drag the word into the place where it should go, or you click and click in the column and it'll go where it should go, and all right, so you want to do that. So this is practice. Quiz questions. Make sure you can answer those questions. So if you say you're done, you're not done. You need to come back and do this review. Remember, you have a test next week. So you are reviewing. Use all the time you can to review. Remember, the test also covers Introduction to Life Science. It covers macromolecules, and it covers cell transport. 3 modules. All right, and I had somebody, you don't have to, if you need tutoring, extra help, you don't have to sign up on the website, on the date book. I'm just here in the Zoom on Tuesdays and Fridays, 1:30 to 2, just show up. I will be there. If I'm talking to somebody else, we'll make space. All grade, anything that you need to make up needs to be completed by, I'd like it completed by Friday, but most definitely by next Friday, because that's the end of the grade term. So after that, you can't make up any zeros. They stay zeros, and we work on next semester. Jamarion, I'm going to create a breakout room, and we need to talk. All right, so I've sent you an invitation to the breakout room, Jamarion. So let's meet me there. Okay. I'm back. Close that. All right. All right. Avenue. Okay, Avenue, I'm inviting you into a breakout room. So join me in the room, please. All righty. You know, I hope everybody's good because I forgot to open Go Guardian. Malaysia, you good? Because I know you were absent yesterday. All righty, Monty, where did you go? Yes, that is what we're working on, cell transport. Wow, and time has been going very quickly today. Folks, it is time to go. You all have a great lunchtime, and I will see you tomorrow. No, I want to see you Friday. See you to Friday. Oh, it is not too early to show me that. Thursday.